Photosynthesis, day 5, the Calvin cycle. Also known as the dark reactions, also known as the light independent reactions, but we don't like those because Calvin cycle can happen whenever it wants, as long as it has the inputs and outputs. They called it the dark reactions to differentiate it from the light reactions, but that caused a lot of confusion. People thought that the light reactions only happened in the daytime and the Calvin cycle only happened at nighttime. But in reality, the light reactions do only happen in the daytime, but the Calvin cycle can happen whenever it has the inputs it needs. So, the Calvin cycle uses the energy from ATP and NADPH, which is what we learned about the. Goodness gracious, they're the products, is the word I couldn't think of. They're the products of the light reactions. So we went through the light reactions. We got sunlight, we got water, and we wound up with ATP and NADPH with oxygen as a byproduct. So we're about halfway there. The Calvin cycle uses chemical energy from ATP and NADPH to reduce carbon dioxide to sugar. So we use chemical energy. Ooh, I can't spell today. From ATP and NADPH to reduce carbon dioxide to sugar. And that's our last step. That's how we get from our solar energy to our sugars. We can call this process the anabolic building of carbohydrates. Anabolic because it is synthesizing something. Remember, anaboly or anabolism is building something. Catalysis, catabolic reactions, catabolism is breaking stuff down. So, and together they make metabolism. So, anabolism is building carbohydrates. Well, it's building anything, but specifically here, we are building carbs. Now, there's a trend in biology where you learn something one way. You've always learned that photosynthesis is 6 CO2 plus 6 H2O with light energy, giving us glucose and water. The end product isn't really glucose. We say that because it's easy to remember and we've got easy numbers. But the end product's not glucose. It can be, but it hits something else first. The end product is glyceraldehyde 3-phosphate. Or G3P. Now, this is a 3-carbon sugar. And if you remember, glucose is a 6-carbon sugar, which means glucose is 2 of these put together. It's how we tend to store 3GP. We don't just have 3GP laying around. Glucose is a pretty common storage method for 3GP. But the end product's 3GP. And we get from our carbon dioxide to 3GP in 3 different phases. So the Calvin cycle has a lot of different steps, and we will touch on some of them. But it happens in 3 phases. Phase number 1 is carbon fixation. Carbon fixation is taking carbon dioxide out of the atmosphere. So the Calvin cycle incorporates each CO2 molecule one at a time. Incorporates each carbon dioxide molecule one at a time. So it'll take a CO2 molecule from the atmosphere or from the inside of the cell and attach it to a 5-carbon sugar. That 5-carbon sugar is called ribulose bisphosphate. Or RUBP. We like our acronyms. So the first step, we're going to take CO2 and we're going to take one molecule and we're going to attach it to this RUBP. The enzyme that does this is really important. If you take one like fancy chemical name from this process, it is the name of this enzyme that does this process. The enzyme that fixes carbon is called Rubisco. So Rubisco is in charge of taking carbon dioxide out of the atmosphere and fixing it to this RUBP, which is really, really, really important for the process of photosynthesis. If we couldn't fix carbon dioxide, we couldn't get anywhere. We couldn't make these sugars. And for that reason, Rubisco is the most abundant protein in plants. It's also one of the very few enzymes that we are going to talk about that doesn't end in ACE. So Rubisco, even though it doesn't end in ACE, is still an enzyme. It's the most abundant protein in plants and possibly on Earth. We can't really do the exact calculations, but of the proteins that are made by living things, Rubisco is up there. We need a lot of it. So, we've got our 5-carbon sugar, we've our RUBP, we've got our CO2 molecule, we've got Rubisco. And that gives us a 6-carbon intermediate. Actually, so we've got our CO2 and our RUBP, and we're going to use Rubisco, and we're going to get our 6-carbon intermediate. We call it an intermediate because it almost immediately splits in half. And we get 2 3-phosphoglycerates. So we had our carbon dioxide, we had our RUBP, and Rubisco took those two and smashed them together, and we got this 6-carbon intermediate. And that 6-carbon intermediate immediately splits in half and gives us 2 3-phosphoglycerates. We are keeping track of carbons because that is very, very important. If we lose a carbon, something's gone wrong. So we had 1 carbon and 5 carbon. We got 6 carbons. Now we have 2 3 carbons. And that's step 1. We've got carbon dioxide out of the atmosphere now. Now we have to get it to something we can use. That's where we get step 2, which is reduction. A lot of redox reactions in biology. So each of our 3 phosphoglycerates gets a phosphate group from ATP. And now we have 1, 3 phosphoglycerate. The numbers, when you see like 1 comma and a dash and then a big long chemical name, tells us where in that chemical those phosphates are. It's not necessarily something you need to know now, but just in case in the future, those 1s and 3s tell us where on our glycerate molecule those phosphates are. It's a big organic chemistry naming structure. So if you take an organic chemistry class, that'll pop up a lot. So we've used an ATP and we've put one of those phosphates onto our phosphoglycerate. Now, let's see where we're at. Okay. We're going to have NADPH donate some electrons. Sorry, I'm trying to follow my own logic. So NADPH is going to donate some electrons to this 1,3 phosphoglycerate. And from there, we are going to get glyceraldehyde 3-phosphate. So when NADPH donates those electrons, we're going to lose a phosphate group, and we're going to get glyceraldehyde 3-phosphate. So ATP donated a phosphate group. NADPH is going to donate more electrons. We're going to lose a phosphate group, and we're going to get glyceraldehyde 3-phosphate. So in the process of gaining and then immediately losing a phosphate group, we have gone from 1,3 phosphoglycerate to glyceraldehyde 3-phosphate. We're just moving stuff around. Now, let's see. For every 3 molecules of CO2, we get 6 3GP. So this right here, or G3P, sorry. That's where I got confused. I was like, I've read that word before. I don't think we've gotten there yet. This is our end product. This is our G3P. We're in step 2 and we already have it. So in this reduction thing, we've used an ATP, we've used an NADPH, and now we have turned those 3 carbon sugars, or those 3 carbon molecules, from carbon fixation to what we need. So for every 3 carbon dioxides, we'll get 6 of these 3GPs. But only a net gain of 1. because the rest finish the cycle. So, after step 2, we have our byproduct, right? We have what we need, we have our sugars. But because it's a cycle, we have to get back to the beginning. So step 3, or phase 3, is the regeneration of RUBP. It's Calvin's cycle for a reason. So we have 5 molecules of 3GP. Or G3P. Sorry, I don't know why I keep getting those switched around. G3P. Glyceraldehyde 3-phosphate. Because we lost one. One of those we can use. We got 5 left. And those carbon skeletons are going to rearrange. And we get 3 molecules of RUBP. So instead of 5 molecules of a 3-carbon sugar, we're getting 3 molecules of a 5-carbon sugar. This takes ATP. So we need to put even more ATP into this cycle in order to get us back to the beginning. And from there, our cycle continues. That RUBP can get fixed by Rubisco, get another carbon dioxide, and do the whole cycle over again. Yes. Rubisco is the enzyme that catalyzes the reaction that gets us from CO2 and Rubisco to our 6-carbon intermediate. It fixes carbon dioxide from the atmosphere. And I'll draw this out too, so we actually have the cycle. We're going to talk our net production though. From the net synthesis of 1,3 or G3P, it costs 9 ATP and 6 NADPH. Which, I mean, makes sense. We're not losing a heck of a lot, because whenever we do, if we've got 2,3-GP making a glucose goes through cellular respiration. We get what, like 36 ATP from that. So between the two of these, it'll even out. We lose a little bit, but it's still a great energy source for us. So, ready to draw this out? Okay, we're going to put our input. We've got, let's see. All right, we've got 3 CO2, right? That's a carbon. That's a carbon. That's a carbon. That's how we start. but they're fixed one at a time. I don't know why I have 3 there. Yeah, we've got 1 per cycle. So we're going to take one of those, and with Rubisco, we're going to get our 6-carbon short-lived intermediate. Draw that out. We got 1, 2, 3, 4, 5, 6. From there, we've got our 3 phosphoglycerates. Technically, we have 6 of them. We got 1, 2, 3 carbons. We're just going to say this is 1. So we got 1 of those, and now we have 2 of these. And we got a phosphate coming off of it because it's phosphoglycerate. Then we're going to use an ATP, ADP, and we're going to get 1,3-biphosphoglysceraldehyde, or glycerate, sorry. 1,3-biphosphoglycerate. Then we're going to take 6 NADP, or actually this is 6 up here as well. NADPH. We're going to have NADP plus. And that will give us 6 molecules of G3P. I'm going to come back up here. Wait, where are we at? Actually, so this right here, we're going to say we've got 6 of them because we said that we had 3 of these and 3 of those, just to keep our carbons straight. Sorry. So far, we have taken our 3 molecules of CO2. We have put them into the Calvin cycle with Rubisco, and we have 3 of these 6 carbon intermediates. With those 3 6-carbon intermediates, we're going to break those in half, and now we're going to have 6 3-carbons. We're going to use ATP. We're going to change it into a different 3-carbon. It'll actually look like this. And now we have 6 of our 3 GPs. G3P, sorry. Is ripped off. That's our sugar product. But 5 G3Ps are going to continue the cycle. We are then going to use more ATP. And we are going to get our ribulose bisphosphate, or our RUBP, which is a five-carbon sugar. We're actually going to get 3 of those. And those 3 5-carbon sugars are going to go up and get a carbon dioxide attached to it through Rubisco. That's our Calvin cycle. We've got 3 different carbon dioxide molecules. We're going to add them one at a time. We're going to get 3, because we're doing this 3 times. We've got a short-lived intermediate, a 6-carbon. That's going to break into 2 3-carbons per cycle. That's going to get moved around a little bit and moved around a little bit until we get our 3GP. We're going to have 6 of those per every 3 CO2s. So it takes 3 CO2s to get 1 of them out. That's why we did it in an interval of 3, because if we didn't have 3, we wouldn't have a byproduct yet. 5 of them are going to re-enter the cycle. We're going to use some ATP to get it from this sugar that we use into this ribulose bisphosphate. And that is going to go into the beginning of the cycle again. That was a lot. So, anybody have any questions about photosynthesis? So we learned about all of those photosystems in the light reactions, how we have our special pigments that can take that can absorb sunlight and raise electrons, go through an electron transport chain. That electron transport chain will help us make ATP. That ATP will come into here. That's this ATP right here. It'll go through a second photosystem, go through a second electron transport chain, and get NADPH, which is this NADPH right here. Then we're going to go to the Calvin cycle, where we take carbon dioxide out of the atmosphere. We're going to use NADPH and ATP, and then we will get NADP plus and ADP. ADP and NAD plus will go back to the light reactions, so we can reuse them. So the light reactions and the Calvin cycle depend on one another. Not sure if we go back to our first day. Yes. If we go back to our very, very, very first day on photosynthesis, we had our big old chloroplast. We got our 2 inner membranes. Our thylakoids. A lot less. A lot neater than that. But we've got our light and our water coming in, and we know exactly where that light and that water goes, which is to that thylakoid membrane. We're going to split that water to steal some electrons. When we split that water, we're going to have oxygen come out. And we're going to use those hydrogens to build up a concentration gradient. When we build that concentration gradient up, we get ATP. And we're going to send the electrons from that light through a couple of electron transport chains, and we're going to get NADPH. And both of those will go into the Calvin cycle, which will take CO2 out of the atmosphere And now we know there's a whole big long process of adding those ATPs and adding those NADPHs and moving carbons around until eventually we get our sugar, which we know is a G3P molecule. Or some form of CH2. That's a very, very crash course way of doing it. There's about 10 minutes left. There's an exit ticket on Canvas for you to fill out. And then tomorrow, we're going to do exactly what we did with the photosynthesis kind of wrap up. And you're going to get as detailed as you possibly can, draw out the process for me, just so we can see it. So we're going to compile all of the diagrams we've used and track all of those carbons and all of those electrons.